Hello, everyone, and welcome to today's webinar titled Keynote Presentation, Viromics, Lessons from Ocean, Soil, and Humans. This webinar is part of the 2021 Microbiology Virtual Week, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on the Ask a Question box located on the far left of your screen. And as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Matthew Sullivan, a professor of microbiology and civil, environmental, and geotic engineering from the Gordon and Betty Moore Foundation, investigator, and he's the co-director of OSU Infectious Disease Institute and Microbiome. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Sullivan, welcome. You may now begin your presentation, sir.
Thank you so much, Susie. It's great to be here. My lab studies how ocean viruses impact marine microbes, and we do this to understand how the oceans work, how they're going to be impacted by climate change, how they may cause climate change. But we also do this because oceans are complex communities. And this means that by pioneering technologies and approaches in the ocean ecosystems, we can get a roadmap for how we can apply and understand virus studies in a complex system. And we can use that then as a roadmap to study viruses in other complex systems. These approaches are generalizable. So here's where we're going today. First, why do we study viruses? And particularly, we're currently focusing on viruses of microbes. In fact, double-stranded DNA viruses of microbes. Then I'll spend a lot of time focusing on the oceans under the themes of patterns, processes, and paradigms. And then we're going to talk a little bit about what we've learned studying viruses in soils and in humans. So why do we care about viruses of microbes? I often say you only care about a virus through the host that it infects. Now, viruses that infect humans and human cells, like the current coronavirus world that we're living in, are obviously important. They impact health. But microbes in your body are as abundant as your human cells, and there's 100-fold more microbial genes in your body, and those microbiomes in your body are increasingly recognized to impact human health. Microbes in soils are also important. Did you have cereal for breakfast this morning? Microbes that live in soils help make the nutrients that feed the plants that you ate. Microbes can help improve crop yields. And if you just breathe the breath of air, half of the oxygen in that breath came from microbes, ocean microbes, or tiny plants in the oceans that produce half the oxygen on the planet. Now, my interest is more focused on viruses. These microbes are important. Could viruses infect them? And if so, what role do viruses play? We're going to start in the oceans. And in the oceans, we actually didn't think there were many viruses. People went out with cultures like E. coli and tried to isolate viruses and didn't get any. However, when we concentrated seawater, such as shown here, and stained it with a cyber green DNA stain, we saw what I call stars against the night sky, where these big dots are cells and the little dots are viruses. And so these are big numbers. If you swallow a mouthful of seawater, there's actually 50 million viruses. Now, don't worry, they don't infect your body or your human cells. They don't even infect your microbiome. They're really, of course, designed to infect microbes that live in the oceans. When we do tracer experiments to understand their impact, we learned that about 1 out of 3 cells in the oceans are killed per day. And a challenge in our science is that most of these viruses are completely unknown to science, let alone can be cultured. And when we do other kinds of experiments to look at gene flow or viruses moving a gene from one cell type to another cell type, like antibiotic resistant genes, for example, we see that there's a huge number of genes moved per day, 10 to the 29th genes moved per day. In fact, these virus numbers in the oceans and on the planet are so big, there's thought to be 10 to the 31 viruses on the planet. And if you put these tiny viruses end to end, they would stretch light years away from Earth. So these are really big numbers. When we think of microbial toxins like Bordetella toxin, cholera toxin, Shiga toxin, pertussis toxin, it's not really the microbe that encodes that toxin. It's actually a virus integrated into the microbial genome that encodes the toxin. So it's understanding these viruses and how they work that's really critical for so many ways that the oceans work as well as other ecosystems. But it's been a big challenge to assess these ecosystem impacts because we can't do things like grow the viruses and culture them. So we're going to stay in the oceans here for a while, themed on patterns, processes, and paradigms. And I have a picture of Earth just to emphasize the oceans dominate Earth. 70% of the surface area of Earth is the oceans. And these oceans that we've studied, we've done globally. So focusing on these patterns, I want to introduce the Tara Oceans Consortium, which is a many different principal investigators, international, and we sought to study in the top left the global oceans. You can see the cruise track throughout the global oceans using standardized approaches in the top right, which include literally a sailboat. We'll see a picture of it in a minute that has all kinds of instrumentation measuring down through the water column and specifically looking at both imaging and sequencing data. And then at the bottom, you can see we studied 8 orders of magnitude of ocean organisms, from viruses to fish larvae. This is really a large scale to try to capture biologically and globally. And this crazy adventure has been going on since 2008. And on this sailboat, this is the Tara, and this sailboat has been retrofitted for both below deck work that we have to work with sensitive instrumentation and above deck work to process seawater samples as they come on board. So this is an amazing research opportunity to get global samples, and we're interested specifically in viruses. Problem is, as soon as we start to study them, most of what we observe is unknown, completely new to science. I'm not talking about sort of a new variant within one particular species of viruses. I'm talking about completely new to science. And in fact, we had to develop a set of analytics to handle this. So when we started, we had to just start by cataloging viruses. And here's a picture of where we were when we started. There were 2,000 genomic sequences for viruses and microbes. When we finished some of our early work, we had augmented those databases from 2,000 to 30,000. And in a recent paper, we augmented that 100-fold to 200,000 virus species. And these are many, many more genotypes within these variants. So we had to develop analytics to be able to classify these as species, and I'll mention more about that later. We also had to develop machine learning analytics to actually handle genome-based viral taxonomy. And that's now a gene sharing-based network approach that's established as one of the modern ways to look at scalably and systematically virus taxonomy. So we've got some catalogs now, hundreds of thousands of viruses cataloged from around the oceans. What do we start to see? And so this is our cruise track where the gray dots are where the boat stopped for 24 to 48 hours and did a bunch of oceanographic sampling. And the arrows between the dots represent the movement of virus populations based upon abundances of the genomes, genome-enabled virus tracking. And what we see when we layer on the ocean currents is that these viruses seem to ride or drift with these ocean currents. Now, this is not all that interesting, and I'm going to come back to it later, but it's kind of exciting to think we can use genomics to be able to track the flow of viruses through a complex system as large as the global ocean. Beyond genomic tracking, we can also just simply identify who are the viruses that are there. And so we've done that pretty extensively at this point. And one of the most abundant viruses is a virus called T4. And many of you have probably heard of this virus because it's the basis of most of the molecular biology reagents that are used to do fundamental laboratory experimental manipulations. T4 was studied for decades now for its molecular biological properties, and for that reason has very useful enzymes and reagents for molecular biology. Another character, though, I'd like to introduce is Phi-38.1, and most of you have probably never heard of Phi-38.1. I hadn't either. We happened to get it into culture, and we'll see some of the work that we've done with Phi-38.1 later to make it one of the most well-characterized viruses on the planet, and here the third most abundant virus genus in the global oceans. So these kinds of global ocean patterning data, just very simple descriptive information of who is there, allow us to create diversity anomalies when we place this information into predictions about what the oceans will look like for models. And that allows us to plan things like, how will fisheries catches change in response to climate change? How should we set up marine protected areas so that we get the biggest bang for the buck for where we're trying to preserve areas of the oceans? And so that's very valuable applied aspects of this very simple first description of what viruses, who are the viruses that are there, and what are their abundances like throughout the global oceans. The next vignette within oceans I want to focus on is processes. So how are viruses impacting processes? And before tar oceans, from cyanobacterial viruses in culture, we had learned that viruses impact processes in the cyanobacterial system through metabolic reprogramming using auxiliary metabolic genes, or AMGs. And in particular, they stole the core photosystem reaction center genes. So this is what we think is happening. This is a photosynthetic membrane in a cyanobacterium, for example, where we have two photosystems, PS1 and PS2, embedded in the photosynthetic membrane. And a photon of light would strike the first photosystem, excite an electron, pass that through a series of electron donors to the second photosystem, where a second photon of light would strike, excite another electron. That electron would be passed to ferret oxygen and on to other reactions. The viruses almost all steal the core photosystem 2 reaction center protein called PSBA. Now, this is an important protein because it splits water to make hydrogen and oxygen. So important, in fact, that 4 Nobel Prizes have been won studying this kind of reaction center. Viruses also steal other genes. Here I've highlighted the cognate pair of PSBA, PSBD, as well as highlight inducible proteins, plastocyanin, and ferret oxygen. These, we think, are drawing electrons away from the PS2 reaction center to keep it going. And I forgot to mention the PSBA protein, because of its proximal to splitting water into oxygen and hydrogen, actually creates oxygen radicals and turns over on the scale of 30 minutes in every photosynthetic cell. So the virus really is encoding genes which help keep this photosystem in place, and in particular might function with respect to rate limiting steps that feed back to making more viruses. So we think of this as viruses probably randomly steal genes from their microbial hosts. And that makes sense because we know viruses tend to mispackage DNA for many reasons. And those genes which have a fitness advantage here in a cyanobacterial photosynthetic cell to keep photosynthesis going, they could be selected for in that virus genome and kept around. So these virus photosynthesis genes are important for these cyanobacterial viruses which need photosynthesis during their infection. So what do we learn from Tara oceans? Is photosynthesis the only thing out there? No, not at all. In fact, there's lots of central carbon metabolism genes scattered across viruses throughout the Pacific Ocean and other global oceans. There's sulfur cycling genes and nitrogen cycling genes scattered throughout virus genomes in the Tara oceans data sets. So there's lots of ways that viruses have collected key metabolic genes and alter the biogeochemistry or the processing of these key nutrients in the oceans. And another interesting element is, which I won't show, is that we've actually looked at the evolutionary history of some of those photosynthesis genes, in particular PSBA and PSBD, and shown that the virus version evolves in the virus under different selective constraints, and part of that can be put back into the cyanobacterium, altering the photosynthetic trajectory of the cyanobacterium. So there's a real interesting interplay between viruses and their own fitness advantage and the coevolutionary history of these cyanobacterial cells. So we're going to walk through 3 paradigms that I think are new ways we think about how the oceans work as a result of our Viromics contributions. And the first one is focused on the idea of virus sequence space. For my research, I would like to be able to identify viral populations in environmental data. And the problem is that virus genomes are typically thought of to be rampantly mosaic or have some sort of genetic continuum, akin to like the quasi-species concept in RNA viruses. So if we just sort of pictorially look at this, on the left would be a scenario where gene sequence space of the yellow viruses blends into that of the green viruses, blends into that of the dark green viruses, blends into that of the blue viruses. So where would you actually draw a line? Is there any discreteness in this genome sequence space to where you could actually draw a line about biological entities? And the right case, which we would hope for, would be that yellow viruses exchange genes maybe across these populations, but that there's a big gap between the genome sequence space of the yellow viruses from the blue, from the green. And so we were interested in asking, okay, the paradigm would be the left for viruses, but it turns out that really the paradigm for what genome sequence space might look like is driven by fast evolving viruses, single-stranded DNA and RNA viruses. We know that double-stranded DNA viruses evolve much more slowly. Could it be a different story? And so if we go out and isolate viruses from the oceans, I spent 10 years doing that in my PhD and postdoc, you get this as a representation of cyanobacterial virus genome sequence space. So these gray dots are 10 years of my efforts on many hosts and many seawater samples to isolate and sequence genomes. We invented a new method. So then when we went and isolated genomes from one seawater sample, we got the blue dots, which you'll see are almost as spread from 4 genomes as all of that I found all over the world's oceans with many different hosts. And then we invented a method that could look at 10 to the 7th viruses in a 10-minute experiment. This is called viral tagging. And this led to the red dots, where we saw all of sequence space covered, and we got variation within each of these populations. And what you can see is that in no case does the variation, the cloud of reconstructed population genomic sequence space, overlap with another cloud. And where you think it does, if you look into the third axis, it's the end of the page axis that is separating them. So this suggests that in a wild seawater sample with a single cyanobacterial host, that there's population structure. And when we do follow-on experiments that specifically looked at using a population genetic metric of biological species, we saw from 142 complete cyanophage isolate genomes from these same seawater samples that these are consistent with a species. Gene flow is higher within and between these groups, differential population selection, and these are fixed genomic features. As well, we can look in other systems outside the cyanobacteria and see that the cutoffs, this 95% ANI or greater than 5% ANI divergence, which is average nucleotide identity, those cutoffs are a good cutoff not only for cyanobacteria, but also for heterotrophic viruses, where we did phenotyping with a pseudoarthromonas phage. Now, beyond species, as I mentioned earlier, there's the gene sharing networks. And these we can get at genus level taxonomy using that machine learning approach. So this is helping us rethink about virus sequence space. What about the ocean carbon pump? And you should care about the ocean carbon pump because it turns out half of the carbon dioxide that we humans put into the atmosphere is absorbed by the ocean. And when the ocean absorbs all that carbon, it actually interacts with water through germ chemistry to become acidic. And we've shown now through decades of just monitoring in both the middle of the Pacific Ocean and the middle of the Atlantic Ocean that humans have successfully acidified those massive ocean basins of the Pacific and Atlantic Oceans. And that's happening due to increased CO2 diffusing from the atmosphere into the oceans. So for decades, we've been interested in how the ocean carbon pump works. And I mentioned earlier that our genomic tracking result was not that interesting. Viruses don't swim. And so, of course, they ride the ocean currents. What I didn't show you is that there's also X's at each of those, at many of these sites. So the X is an example of viruses sinking. Now, viruses have a low Reynolds number, so they shouldn't sink. How could viruses be sinking? And the paradigm in our viral ecology is that viruses in the oceans would infect bacteria and phytoplankton, and this would lead to cell lysis, which can result in particulate and dissolved organic matter. It then becomes food in a very nutrient-starved surface ocean for being recycled by those same bacteria and phytoplankton populations. And this is called the viral shunt. And the idea is that viruses should keep carbon small and prevent carbon from sinking. But an alternative idea was proposed by Marcus Weinbauer that you could actually get these localized particulate organic matter could stay very localized as aggregates, and those aggregates are deep enough to sink out of the water column and could contain sticky viruses in them. So that could lead to carbon export. As well, viruses, when they infect cells, maybe preferentially, virus-infected cells may be preferentially grazed, and there's actually data both for and against this, depending on the model system you look at. So, huge question, are viruses leading to altering the ocean's biological carbon pump? And we decided to test this with what we call genes to ecosystems modeling. And the idea is that you have lots of sequence data telling you what the organisms are and where. You run those through machine learning analytics and ecosystem modeling analytics, and you ask, what is the best prediction for the data observed? And in our case, we were interested in the sailboat's trajectory and our estimate of carbon flux, which is pictorially represented here down into the water column, where red are high areas of carbon flux and blue are low areas of carbon flux. And we were interested in the simple question. Remember, Tara Ocean studied viruses to fish larvae. So which of the organisms in that spectrum drive carbon export in the oceans? And so we applied these machine learning and ecosystem modeling analytics, and we found that viruses, better than any other organism, in fact, could explain 89% of the variation in carbon flux, which in ecology, which is in complex communities, you never explain 89% of the variation of anything in nature. That's a really big, strong signal. And so it really suggests that viruses are forming these aggregates and leading to carbon export, as indicated by the X's in that earlier plot. Now, some of you still don't perhaps care about oceans and carbon export, and I totally appreciate that. But this machine learning and ecosystem modeling analytic is generalizable. So I could have been in another system and looking at thousands of microbes or viruses and asking questions about which ones best predict disease or soil health or product yields. And what I didn't tell you is we don't just know from this earlier study that viruses best predict. We actually can use leave-one-out modeling to figure out which ones are the most important ones. And so we actually know sort of the top 5 viruses. So out of thousands, we know which few to focus our studies on. So the third paradigm I want to reach out about here is phage resistance. And the idea here is that phage resistance is simple. Here we've got a cell with its genome represented in the inner circle, and we've got a virus which might infect it. So adsorption is the first step resistance could occur. And then the secondary step is that maybe the virus can get its DNA inside, it absorbs, but there are defenses like CRISPR, abortive infection proteins, or restriction modification systems. And the third is that in the oceans, a virus may not find the right host, so it finds an alternative host, and it may just simply not have the right molecular machinery to tuned to be able to make the takeover process efficient. So mismatched utilization of the host machinery. So the idea here is, in the lab, if we can make a genetic mutant, that's a very abrupt change, targeted, and so single changes are found to drive resistance. And we wondered, is this also true in nature? And so our effort to set up an experiment goes like this. We had one phage which infected a Bacteroidetes host well and another alternative Bacteroidetes host poorly. The two Bacteroidetes had identical 16S sequences, but were isolated from water such that background mutations around the genome had occurred. And the virus is this fellow I mentioned earlier, 538.1, which is the third most abundant ocean virus genus globally. So a very ecologically important virus. So again, it infects one host well. We see clear plaques. We see a nice one-step absorption curve. We see phage production after 80 minutes. And the same virus on a 16S identical host sees terrible plaques, no clear one-step, and no obvious phage production after 4 hours. We did some work with a single-cell molecular probe technique we developed called phagefish, which shows that this is inefficient infection. It's not just a lysogenic infection. And so we asked the question with time-resolved multi-omics data sets, what is happening? Why is this alternate host strain inefficiently infected? And what we found was, first, that yes, absorption is part of the problem. 30% poorer absorption based on absorption kinetics experiments. We also found, however, that fewer cells were successfully infected just due to failure to block restriction modification systems. And this was data we were able to infer from both phagefish and the multi-omics data set. Again, we found more evidence that the host defended against this infection by both degrading phage DNA and proteins and stalling their ability to make phage DNA and proteins. So this is really different than the abrupt single mutant change. This suggests that what I call nature's mutants, identical 16S sequences, cells isolated from the same water column but have slowly drifted. Nature's mutants reveal resistance to phages is not simple. Here, there's at least 4 ways that this cell is being inefficiently infected. Now, to complement this kind of work, we also do work to understand what are called virus cells. The idea that the virus-infected cell is quite different than the non-virus infected cell, and trying to characterize that using time-resolved multi-omics data sets. And as well, we're interested in how virus cells respond to nutrient deprivation and to grazers. So hopefully there's a picture at this point that that even though I just study viruses, we try to really do it in a broad ecosystem context, and that's really powerful. Here, for example, if we were just focused on the leg of this elephant, we might say it's a tree. Or if we were just focused on the tail of the elephant, we might say it's a rope. But obviously, where we're all sitting, where we can see the whole elephant, we realize it's an elephant. And I'll just emphasize that Tar Oceans is really pioneering and leading many of these integrative approaches. And also at Ohio State, where I'm at, we've got some really amazing institutions in place now with the Center of Microbiome Science, which is 80 faculty across 9 colleges, sharing and creating resources in microbiome science tools, and a new NSF-funded Biology Integration Institute called eMERGE, which we'll show a little bit of data from here in a moment. Now, if you're not at Ohio State and you want to study viruses, we've also tried to develop tools that will democratize the capability of doing this virus microbe Viromics. So this iVirus tool is at iVirus, and that's the workflow pictured in that figure. This iVirus compute is free, provided by NSF. Anyone in the world can use it. We've had extensive documentation on the live protocols at protocols.io. This allows the community to tell us when we've gone wrong and give us feedback on what's working and not working. We also have put out a number of benchmarking papers where we're recognizing issues that are coming up in the field, people not knowing which tool to use for a certain step. And so we've done benchmarking analyses to provide guidance for the field. to make it easier and easier so students don't lose months and months of time troubleshooting things that should be worked out. And we've held a number of Viromics workshops, training hundreds of people from around the world in these years already. And our next one is, I forgot to update this, October this year, October 2021. We've delayed it since last October, and it will be virtual. So that's iVirus. So that's the oceans. And I want to shift gears for the last part of this talk to very simple vignettes in the soils and in humans. So our virus is important in soils. And there was a recent review paper that looked at this. There are known to be a lot of viruses that are abundant in soils. But a recent review paper suggested if we look at all of the features like physicochemistry and down here viruses that might structure bacterial communities, it was proposed that predation and viral lysis is the least important of structuring microbial communities. Now, in the same review article, there was a section entitled Viruses and Their Role in the Soil Microbiome, which basically said, but I guess we don't really know much at all about it yet. And so maybe placing them on this spectrum isn't so refined yet. But we were interested in whether this might be the case. And so by we, I'm part of a soil consortium that was called Isogeny and involved a lot of PIs that are listed here. And this Isogeny project grew into that eMERGE Biology Integration Institute that I just mentioned. So we're interested in a model ecosystem in Sweden called Stordalen Mire. And this has been a long-term ecological research station in a very beautiful location up in northern Sweden, and heavily instrumented to be able to look at lots of ecosystem science-related fluxes and measurements of interest. And it is an area of active thaw. And so we looked at the field site and identified essentially three classes of thaw. This is a natural thaw gradient where we had some areas that were dry permafrost pulsa, some that were intermediate thaw that were sphagnum-dominated bog, and then some that were fully thawed or diaphragm-dominated fen. So from this permafrost thaw, we'd sequence hundreds of bulk metagenomes to look at their microbial signatures, and out of that, we mined viral populations, nearly 2,000. So we're seeing viruses. First of all, they're clearly present, and those numbers are getting quite high. We've doubled known soil viral genera. Most of these viral populations appear to be active. A big question in soils is whether viruses are just inactivated or relic in response to soil and physicochemical interactions with soil particles and minerals. Beyond them being active, it looks like they're predicted to infect a lot of the key carbon cycling microbes, and they can even encode carbon cycling auxiliary metabolic genes like the photosynthesis story I mentioned for the oceans. So it seems like soil viruses should be playing important roles, and we're now trying to build this new biology information into predictive models. And so to wrap up with the humans focused work, we'll spend a little bit more time here, and with the emphasis on a lot of the capabilities we've developed in the oceans really help in the clinic. Specifically, first, just by establishing quantitative surveys and the ways to actually look at the data. How do you count? How do you model? Because it turns out studying viruses is different than studying microbes or microbiomes. Second is augmenting databases. In the oceans, we've augmented the databases 100-fold. And so just to give you an example of that impact, we applied our oceans approaches to establish a very simple idea, the Gut Virome Database. And in this, we basically took all published studies, 32 published studies of virome data and then microbiomes that have not yet been mined for viruses, 2,000 people, 2,700 metagenomes, some bulk metagenomes, some virus-like particle enriched. And out of that, we identified 33,000 viral populations, which is a species level designation. So a single paper presenting 33,000 viral species. In this, it turned out that it didn't matter really when we did a meta-analysis whether you were diseased or healthy. It mattered more whether the samples were processed in lab A or lab B, because the methods being used across these different papers were so different that the methodological signal flooded the disease status signal. And so this is something in the oceans that we've worked hard for about the last 15 years, developing systematic community consensus approaches so that we have quantitative systematic comparability across sample sets. And so we made that recommendation that this signature is a problem. And then we also did learn some of biology. We learned about the kinds of viruses and the relative richness and how richness changes in the course of the human life, looking at the impact of aging on the viral communities. So that's sort of in the human setting, particular studying viruses in relation to aging, viruses of microbes in relation to aging. We could also, as we talked about earlier, look at resistance, and this might enable really a 100-year-old idea of phage therapy, the idea that a virus can infect a cell and might lyse it and infect another cell and may not lyse it. Why? What are the mechanisms for that? And so we would care about this because there are superbugs or multi-drug resistant, antibiotic resistant bugs, and a number of them are listed here. And so as an alternative to antibiotics for these drug resistant, antibiotic resistant bugs, you might use phages or viruses that could infect this particular superbug and kill it. Where does viral ecology come in? Well, we actually have, because of the complexity of natural systems, we're always looking for scalable, high throughput approaches. And we've got lots established in genetics, genomics, capture and characterization methods like viral tagging that I mentioned earlier, and linking viruses to hosts. So viral ecology can really, I would say, change the speed at which we can enable and empower phage therapy. So there's 3 ways that viral ecology can help. A 4th way is simply this idea of ecosystems level understanding. And here the hypothesis would be that many complex diseases have microbial or viral underpinnings, where the ecology of those interactions might matter. And just as an example, I'm going to talk about autism. Here, we have a connection between autistic children's symptoms behaviorally in the clinic with their GI symptoms. They often have a lot of GI gut problems. And when people studied the gut as a result, they found that bacterial diversity decreased, and particularly fermenting bacteria tended to decrease in the autistic gut. And there was no information on viruses. And so we collaborated with Greg Caporazzo, Jim Adams, and Rosie Kromalik-Brown to try to understand in a controlled pilot open label study. So this is a very small study. We tried to understand how FMT, fecal microbiota transplant, in a sustained manner over 10 weeks could impact symptoms. And what we found is that the GI symptoms decreased quite significantly, and the number of children with autistic symptoms in the clinic were behaviorally recategorized and as improved. And we now track the virus and the bacteria that correlate with these improvements. And so with this kind of as a baseline, you'll remember earlier I mentioned these ecological machine learning and ecosystem modeling approaches can take the thousands of microbes and viruses we see in the gut and identify the few that matter. And then you can do follow-up work on those few. So it's a great first approach to understand which microbes or viruses should I be studying. And beyond autism or aging that I've already mentioned, we've got projects in phage therapy. We're looking at how smoking impacts the lung microbiome and virome, as well as spinal cord injury and how that impacts and is impacted by changing microbiome virome. Those are all published. In addition, we have several ongoing studies looking at other human comorbidities or diseases. So in summary, I view viruses as nanoscale entities that are of global importance, so here on top of the world. And I've tried to share with you the idea that we've developed really good systematic quantitative sampling approaches. I didn't mention it, but we can work with almost nothing. These are quantitative approaches down to 100 femtograms of material, which is something like 7 orders of magnitude less than is recommended for standard library preparations. So we've got really well-characterized, systematic, sensitive, and quantitative approaches. We've used these in the oceans to establish a global catalog of the viruses that occur. We've established machine learning analytics to be able to do genome-based taxonomy, because virtually everything we see is new, hundreds of thousands of new virus species in a single paper in the oceans. And then we've used these information from these virus catalogs and their abundances and their ecologies in genes to ecosystems modelings and in the oceans that was specifically to show that viruses drive carbon flux, supporting this hypothesis that virus aggregates form, and that leads to virus sinking and carbon export in the oceans. We've also applied these kind of methods in soils, in particular a thawing permafrost gradient, to be able to look at how soils, as they thaw, may impact climate change. And in humans, we've been using these approaches both in phage therapy contexts, as well as in trying to look at microbial and or viral underpinnings of complex diseases. I also mentioned that Ohio State's an amazing place right now due to the Center of Microbiome Science, which is big and broad and very focused on cutting edge training, and the Emerge Biology Integration Institute, which provides a great multi-omics data integration platform for studying climate change that could easily be generalizable to other complex systems. And this kind of work involves lots and lots of people. I'm highlighting particularly the trainees as I went along, and again here, who had contributed or driven much of this work, as well as several consortia and sequencing partners for this large-scale kind of efforts, and the funding agencies. Thank you.
Dr. Sullivan, thank you for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's take a look. We already have some questions coming in from our audience. Dr. Sullivan, are you studying all viruses or particular kinds of viruses?
Yeah, so we're currently, the approaches that I described here today are related to double-stranded DNA viruses and focused on viruses of microbes. The methods these days are also applicable to single-stranded DNA viruses, and we're working on methods for large-scale RNA virus surveys.
Thank you so much. And one audience member wants to know, can we use viruses to help mitigate climate change?
Yeah, I think 5 years ago, I would have said no way, and I worry about that. But I'm also growing increasingly concerned that climate change is a problem humans don't want to recognize yet. So if we're to think about the impact of climate change, and we're to think about whether we could understand the oceans to the point to engineer them to help us fight this, worldwide problem, I'm kind of beginning to think, I want to say yes, that we could use viruses to help us mitigate climate change. I'd like to see a little bit more work done in a few key areas where the obvious place you set up large-scale experiments in this space, but I think a lot more. I'm in an engineering department now. I think a lot more about engineered solutions to complex problems like these.
Thank you so much. And again, I want to thank our audience for these great questions and comments. You definitely have some fans in the audience today who are commenting about how wonderful this presentation is. We have one more question coming in. Where do I go to find tools and resources that you've described here, Dr. Sullivan?
Yeah, so all of our work is open access and so available for the computationally advanced person through GitHub repositories and the like. But we also have built out a lot of these analytics on iVirus, which is this platform on the NSF-funded cyber infrastructure. And so there's a lot of resources there, and there's a lot of tools, and workflows are well characterized in some of the papers. So we hope it's helping democratize everybody's ability to see viruses in any complex data set, sequencing data set. And if you have suggestions for how to improve that, we're always looking for feedback.
Absolutely. Dr. Sullivan, it's been a pleasure. Thank you again for your important presentation and your incredible research. Would you like to provide any closing remarks for our audience before we go?
Just to look out for that, our Viromics workshop that will be a little different this fall, next month, is going to be focused on 3 key areas we see as a bit of a mess in the field right now. We're going to have 3 experts in each of those sessions. So that'll be a nice opportunity for the field to engage. And if you're new to the field, to kind of learn some new things, to not lose time on that research. And please let us know if there's any bottlenecks in your own work. We're constantly trying to help people out there do this kind of work much easier.
Absolutely. And again, I want to thank you for your time today. Before we go, I also want to thank our audience for joining us today and for their interesting questions. And any questions we did not have time for today, and those submitted on demand period, will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 9th, 2023. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care everyone and be safe. Bye-bye.